Welcome to our channel. In this video, I am going to discuss about the transposons. Transposons are otherwise called transposable elements. These elements can cause a mutation effect in the genome. Many classes of transposons are available. So let us see one by one what are the application and what are the classes of transposons like this. So the genetic elements that can jump to the different locations within a genome. So that's why it is called jumping genes. So they are always maintained in an integrated site in the genome. So in first time the transposons actually discovered by the lady scientist Barbara McCLINTOck. She founded these jumping genes on the maize. The spotting on the maize kernels shows the activation of the DNA transposons. Actually, she got a Nobel Prize for this invention. In addition, most transposons are eventually become inactive and no longer move. She got the Nobel Prize for this invention in the year of 1983. Since McCLINTOck discovery, three basic types of transposons have been identified. Class 2 transposons, and next one is class 3, and the last one is retrotransposons. Here the class 3 transposons, otherwise called miniature inverted repeat transposable elements. So let us see one by one these types of the transposons. First we are going to see about class 2 transposons. Class 2 elements are simply segments of the DNA that move from one place to another via the cut and paste mechanism. For example, we are doing some works in the computer system. We have to cut some of the content from that particular page, and again we have to paste that particular content in some other pages. Like these genes are cut from one place and it is pasted to the another place. So this is what I call a cut and paste mechanism. This process requires an enzyme called transposase. So this encoded within some of these transposons. The transposase actually bind to the particular region where the gene is actually cut by some segments, cut into some segments, and even these transposers actually cut the segments where the gene is have to pasted. So both ends of the transposon, which consist of the inverted repeat sequences, that is identical sequences, they are reading in the opposite direction. So the sequence of DNA that makes up the target site, some transposers require the specific sequences as their target site, but others can insert the transposons anywhere in the genome, not specifically but with any part of the genome sequences. Some sequences have the target site, some may be pasted to the anywhere in the genome. So this picture representing how the transposase bind with the particular region and how it is inserted into the particular genome region. So transposase which acts to clear the ends of the transposon, freeing it from its initial location in the genome and clears the target site where the element is to be inserted. You have to see the picture representing here. So this is a cut and paste mechanism. Here the red color region of the segments are cut with a particular region, then it is inserted to the another region. So you see the second picture, the transposons actually indicated with the violet color. These transposons inserted to the specific region, host DNA region, that the host DNA region representing by the red color, A, T, G, C, A sequences. So it make the split and sticky and cuts. Actually here you see that how the transposase enzyme cut the particular segments in the host DNA. So once it made the cut, the transposon inserted in between the host DNA sequences at the end of the sticky end cuts. So after that the enzymes are involved here to fill the gaps. How the gap is filled here means just see the segments A, T, G, C, A. So automatically the complementary sequences are formed here for A, T. For T sequence, A. Likewise at the both end of the transposons, the new sequences are formed here. So So these class 2 transposons range in length from 1000 to as many as 40,000 base pairs. So this is about cut and paste mechanism. And next one is class 3 transposons, miniature inverted repeat transposable elements. These are characterized by their short length. That's why it calls miniature inverted sequences. Generally about 400 to 600 base pairs, and by a stretch of about 15 base pairs that occurs at each end of each element in an inverted fashion, like a mirror sequence. You just see the sequences here, the 5' to 3' region, GGCCA, GTCA. This is in the first strand, 5' end of the strand have these sequences. In the second strand, the reverse order, these 5' region also have the same sequences like G, G, C, C, A, G. So this is called mirror sequences. At the both end of the 5' have the same sequences. It looks like a mirror sequence. So this is called mirror. In between it have the 400 to 600 nucleotides. But at the end of each strand have 15 base pairs. So this is called miniature inverted repeat transposable elements. The mechanism by which these elements move about genome is not well understood, but thousands of MITs have been identified in the genome of Rhiza setaga and also some of the nematode and some of the other organisms. So unlike some type of transposons, the MITs do not appear to encode the proteins, and most insertions of these elements occurs in the E-chromatin region. So the form of chromosomal material that contains the majority of the active genes. But most probably these MITs sequencers have a role for synthesizing of some microRNAs, miRNAs, which play a role in the RNA interference mechanisms. And the last type of transposon is the class 1. It is called retrotransposons. Retrotransposons represent a highly unique group of the transposable elements. Why it is called retrotransposons means it has some reaction, reverse transcription reaction. How this reactions actually happen, I will explain next. It forms the large portions of the genome of many eukaryotes because the mechanism is a copy and paste mechanism. Because it retains the subsequences in that particular region, and even it copy to the next genome segments. So it makes the paste to the particular genome segments. So in both sequences, it retains the sequences. Both position retain the sequences. So that is called copy and paste mechanism. So they leave behind the original copy and also generate the second copy. So that is inserted elsewhere in the genome. So this process results in the insertion of repeated sequences of the DNA throughout the genome. The mechanism is responsible for the vast spread of the transposon elements in many higher organisms. You just see the picture here. The red color is a particular transposon segments. Even it persists to the particular segments, and even it makes the copy, and also it pastes to the another segments. So see in the picture, the sequences are present in the both region. The first step in retrotransposition occurs when the transposable DNA is copied into RNA. So then the RNA segment jump to the another location in the genome. However, in order to be inserted into the genome at the new site, the mRNA must be copied back into the DNA. So here the enzyme called reverse transcriptase. This enzyme actually synthesizing the DNA which use the particular segments present in the RNA. So this process actually called reverse transcription. So here the enzyme involved here is reverse transcriptase. So these DNA segments, actually these are several different types of retrotransposons have some types like lines and signs. Lines means long interspersed nuclear elements, short interspersed nuclear elements. About 20% of the human genome is made up of the long interspersed nuclear elements. So this is the mechanism of retrotransposons. How the retrotransposons involved in the mechanism means it will copy the segments from the RNA, make the DNA copies from the RNA segments only. So by the process of the reverse transcription, you see the picture here. long interspersed elements. The human genome contains over 5 lakh long interspersed elements, representing some 16% of the genome is about lines only. They are very long sequences. That's why it is called long interspersed elements because 5,000 base pair sequences, DNA sequences that represent reverse transcribed RNA molecules originally transcribed by the enzyme RNA polymerase II. That is messenger RNAs. Lacking introns as well as the necessary control elements like promoters, these genes are not expressed. Because if the genes should be expressed means it will have some promoters and it will have some repressor like that. They are called pseudogenes. But some lines do encode some functional reverse transcriptase and also integrase reaction. This enables them to mobilize not only themselves but also the others. Because this is done by the copy-paste mechanism. So the number of lines can be increased in the genome. Because it retains some sequences in the sequences in the particular position. So the diversity lines between individual human genomes make them useful markers for DNA fingerprinting studies. Short interspersed elements, these are very short sequences. The difference between long and short is the base pair sequence size only. Here the size is 100 to 500 base pairs only. That represent reverse transcribed RNA molecule originally transcribed with the RNA polymerase III. Mainly the molecule of tRNA, 5S RNA and some other small nuclear RNAs. So most abundant signs or AL elements, these are about 1 million copies in the human genome. So they do not encode any functional molecules like lines. Their presence in the genome is mystery. Like lines, they seem to represent only junk or selfish DNA. So how the transposons involved in the mechanisms are causing some diseases. So the functions of transposons remain still unclear. Even though Barbara McCLINTOck found out that the transposons have some mechanisms and it makes some colors in the kernel regions of the maize, still it is remain unclear. They have long been referred to as the junk DNA because they appear to serve little or no purpose, or as a selfish DNA, because they serve only to copy and amplify themselves within the genome. So, but in rare cases, the transposons are associated with the genetic mutations, because if suppose it jump to the any other position in the genome, it will make alters in the genome sequences, automatically to make the alters in the protein synthesis or amino acid synthesis formation. So, the chromosomal rearrangement that cause the diseases in the humans. Disease typically arises from the insertion of the transposons into the particular regions of the genes that are involved in regulating gene activity. For example, the insertions near promoter regions, which are short segments of the DNA that are used to initiate the gene transcription. So, it is well known concept that promoter actually initiate the process of the transcription. It can lead to the overactivity of the genes, but in some cases, this can give rise to the cancer. Overexpression of the promoter may cause some overexpression of the gene, so automatically it leads to rise a cancer. In other cases, the site where a class 2 element is cut out of the genome is not repaired correctly, resulting in the mutation. So that it also interferes with the gene regulation and thereby cause cell dysfunction. So even the cell dysfunction may cause some defects in the genome. So however, they have been found to be the cause of the mutations responsible for some cases of the human genetic diseases. So like these are all the human genetic diseases actually caused by the transpositions of these transposons, hemophilia A and hemophilia B, X-linked severe combined immunodeficiency, porphyria, like these genetic diseases actually caused by the transposons movement. But even though it is used for some techniques or technologies, the uses of transposons are mainly it is used as a cloning vehicle in transformation studies, transformation vector for transferring the genes between the organisms. So especially in the drug resistant gene encoding, by many transposons are useful in the development of the plasmid. because for the drug resistant gene mechanism, we are creating some plasmids. By creating for the plasmids, these transposons are used here. These transposons, even they are used as a cloning vehicle. So the main application of this transposon is it is act as a vector, or it is used for the cloning processes. Like, thank you for watching.